Welcome my dear students. Today we are stepping into one of the coolest navigation systems in biology. In the first part of protein sorting, we have explained how cells use their GPS system. We'll explore how every newly made proteins in our body knows exactly where to go thanks to tiny built-in address levels. It's like the cell's own courier service working with perfect accuracy. Let's dive in and see how this incredible system keeps life running smoothly. Transmembrane transport proteins imported into chloroplast, which is similar to mitochondrial protein import. Protein import into chloroplast is very similar to that in mitochondria. Both processes occur post-translationally after protein synthesis in the cytosol. The process requires energy for translocation. It involves translocator complexes in the membrane. It uses amphiphilic N-terminal signal sequences that are removed after import. However, the protein machinery involved differs between chloroplast and mitochondria. Energy source for import. We know that mitochondria use electrochemical H plus gradient across the inner membrane, that is the in between the mitochondrial matrix and the perimitochondrial space, but the chloroplasts do not have such a gradient across their inner membrane. So instead they use GTP and ATP hydrolysis to drive protein import across the double membrane. Evolutionary relationship. The similarity in transport systems is likely due to convergent evolution, where both organelles independently evolved similar mechanisms to solve the same problem, translocation across a double membrane. Distinct signal sequences. We know though chloroplast and mitochondrial signal sequences look similar, they are recognized by different import receptors. This ensures that each protein reaches the correct organelle. Say for example, a bacterial enzyme attached to a mitochondrial signal sequence goes to mitochondria. The same enzyme attached to a chloroplast signal sequence goes to the chloroplast. This is the structure of chloroplast. We know chloroplast have three main compartments, that is outer membrane, inner membrane, this is the inner membrane, and the intermembrane space, and the thylakoid membrane and space. The stroma is the internal fluid filled space which is similar to the mitochondrial matrix. Many chloroplast proteins such as those for photosynthesis and ATP synthase are located in or on the thylakoid membrane. Two-step protein transport process. The step 1, proteins move from the cytosol through the chloroplast double membrane into the stroma. Step 2, some proteins are further transported into the thylakoid membrane or thylakoid space. Dual signal sequence mechanisms. Proteins destined for the thylakoid have two different signal sequences, an N-terminal chloroplast signal sequence that directs the protein into the stroma, and a thylakoid signal sequence that becomes active after the first one is cleaved off by a stromal signal peptidase. Transport into the thylakoid. Once in the stroma, the exposed thylakoid signal sequence guides the protein across or into the thylakoid membrane. But there are four different routes for proteins to cross or to integrate into the thylakoid, each using different stromal chaperones and energy sources. This is the image showing the protein translocation into chloroplast. We know that the precursor protein contains N-terminal chloroplast signal sequence is labeled as red, followed immediately by a thylakoid signal sequence labeled as orange. The chloroplast signal sequence initiates the translocation into the stroma through a membrane contact site by a mechanism similar to that used for the translocation of mitochondrial precursor proteins into the mitochondrial matrix space. But the signal sequence is then cleaved off, unmasking the thylakoid signal sequence, which initiates the translocation across the thylakoid membrane. Now we should study transport that is proteins transported into peroxisomes, which is another example of transmembrane transport. So the signal for peroxisomal targeting, many peroxisomal proteins have a C-terminal signal sequence made up of 3 amino acids like serine, lysine and leucine (SKL), but some others have a separate N-terminal signal sequence. If either signal is attached to a cytosolic protein, it can direct that protein into a peroxisome. Recognition and import machinery. Import involves soluble receptor proteins in the cytosol that detect the signal sequence. These receptors interact with docking proteins on the peroxisomal membrane. The process requires at least 23 special proteins called peroxines. Energy requirement. Protein import into peroxisomes is driven by ATP hydrolysis, providing energy for the translocation process. Peroxisomal membrane translocator, a complex of about 6 different peroxines, forms the membrane translocator that moves proteins across the peroxisomal membrane. Key differences from mitochondria and chloroplast. Proteins do not need to unfold before entering the peroxisome. Both monomeric and oligomeric proteins can be imported intact. This makes the mechanism distinct from that of mitochondria and chloroplast. Role of peroxine PEX5. The soluble receptor PEX5 binds to the cargo in the cytosol and travels with it into the peroxisome. After releasing the protein inside, PEX5 returns to the cytosol to begin another transport cycle. This resembles how import receptors recycle during nuclear transport. There is medical importance known as Zellweger syndrome, a serious inherited disorder where proteins fail to enter peroxisomes. Cells contain empty peroxisomes, leading to severe damage in the brain, liver and kidneys. The condition is often fatal shortly after birth. One cause is a mutation in the PEX2 gene, which encodes a key peroxisomal transmembrane protein. Defects in receptor recognizing in the N-terminal import signal lead to milder forms of the disease. Now, we will study protein transported into the endoplasmic reticulum. Timing of transport. First of all, we have to know the image showing the transportation from the cytosol to endoplasmic reticulum. The ER captures proteins while they are being synthesized on ribosomes in the cytosol. So there are two major types of target proteins. Transmembrane proteins only partly cross the ER membrane, become embedded in the membrane, and the water soluble protein firstly they fully cross the ER membrane and they are released into the ER lumen. Significance of ER targeting. The ER or endoplasmic reticulum is the entry point for proteins that will be secreted from the cell. They integrated into the plasma membrane. They delivered to the Golgi apparatus, lysosomes or endosomes. So the mechanism overview. Protein translocation across the ER membrane is guided by a signal sequence on the growing polypeptide. So there is specialized machinery including the signal recognition particle and translocator channel ensures proper delivery and their insertion. So all proteins entering the endoplasmic reticulum have an ER signal sequence that directs them to the ER membrane. Two components guide this process. One is signal recognition particle or SRP in the cytosol and SRP receptor on the ER membrane. The SRP or signal recognition particle then binds to the signal sequence as it emerges from the ribosome, causing protein synthesis. So the SRP then brings the ribosome protein complex to the SRP receptor on the ER membrane. The ribosome then attaches to a translocation channel, so SRP is released and protein synthesis resumes. So the growing protein enters or embeds in the endoplasmic reticulum depending on whether it is soluble or membrane bound. Thank you.